Alright, so remember that Chapter 6 primarily goes through and explains all the different eukaryotic organelles and cellular structures. Now we're going to switch gears and talk a little bit more about prokaryotic cells. We know the general gist, right? They're simpler, they're smaller, etc. Let's talk about the structural features that are unique primarily to prokaryotic cells. And this is what Chapter 27 is dedicated to. Keeping in mind there's a lot of material in Chapter 27, and we're not going to go through all of it in conjunction with Chapter 6, right? So we're only going to pick out the salient details in terms of structure, a little bit on function, and then other comparisons between eukaryotic and prokaryotic cells we'll leave for later chapters as we get through to talking about things like cell division, and gene expression, DNA replication, etc. All right, so chapter 27, just a small little chunk of it if you guys are going through and reading your textbook. Okay, so let's review a little bit. We think that Earth's first organisms were likely prokaryotes, right? These are very simple, they're small cells. They are unicellular for the most part, although they tend to sometimes live in colonies. So you'll have individual organisms all sort of bunched together. They're all individual organisms, but they do live together in these groups, which we call colonies. If we were to compare sizes of prokaryotic versus eukaryotic cells, so most prokaryotic cells are somewhere between half a micron or micrometer to about 5 micrometers. That's pretty small. Eukaryotic cells can go from about 10, maybe a little bit smaller than that, to about 100 microns. So that's significantly larger than those teeny tiny little prokaryotic cells. Prokaryotic cells are pretty cool that they can have a variety of shapes, and there are 3 most common shapes that we're going to introduce here in lecture, but that you guys are going to explore a lot next week in lab. So the 3 most common shapes for bacteria are going to be spheres. These guys are called coci. But then we also have rods that look like little cigars or cigar-shaped bacteria. These are called bacilli. And then we also have spirals, which can be called spirilla. If we were to take a look at an example of each one of these, and there are slight modifications. Probably the spheres are the most congruent out of all the different bacterial cell shapes. The rods can sometimes be really long, sometimes short and fat, and the spirals can also have many undulations or maybe just look like an S. So there are going to be slight variations as you guys are looking at different strains of prokaryotic cells under the microscope next week. But so in purple here, we have the spherical ones, or your coci. In the middle, we have the rod-shaped ones, or the bacilli, and then the spiral, or the spirilla on the far right. Okay. An important feature of nearly all prokaryotic cells is their cell wall. Right? We've talked about cell walls before, but when we talked about them, we were talking about plant cell walls. There are some similarities, but in terms of structure and composition, prokaryotic cell walls are very different from plant cell walls. They do share some common functional features, though. So a prokaryotic cell wall is still going to help the cell maintain its shape. It's going to protect the cell, and it's also going to prevent it from bursting in a hypotonic environment. So all very similar functions to eukaryotic cell walls. If we were going to compare, right, eukaryotic versus prokaryotic, eukaryotic cell walls are going to primarily be made of cellulose, if we're talking about a plant, or think back to when we were talking about structural polysaccharides, we also talked about chitin, right? So chitin is a polysaccharide, different chemical structure from cellulose, but it's still an important component of cell walls for organisms like fungi, as well as many arthropods. All right, so that's for eukaryotic cell walls. What about bacterial cell walls? Now, these guys, instead of containing cellulose or chitin, bacterial cell walls are made out of a substance called peptidoglycan. If we were to split up this word, peptido, that probably makes you think of proteins, right? And then glycan probably makes you think of sugars or carbohydrates. And that's exactly what the 2 constituents of this type of molecule are. So peptidoglycan is going to be a network of sugar polymers that are going to be cross-linked or sort of bridged by polypeptide components. Now that's for bacterial cells. Archaeal cells also are going to have a cell wall, but these are going to still contain polysaccharides and proteins, but they lack peptidoglycan, so their composition is a little bit different. We're not going to talk about specifically the composition of archaeal cell walls, but just keep in mind they are different from bacterial cell walls, even though both cells are prokaryotic. Okay, so scientists sometimes have the desire to identify various bacterial strains. Doctors also have the desire sometimes to identify bacterial strains so that they can, for example, treat their patients appropriately when they are suffering from an infection. So there's a very specific technique that was developed by an individual whose last name was Graham, and we call this technique the Graham stain. And this Graham stain allows us to classify bacteria based on their cell wall composition. You guys are going to be able to do a Graham stain in the lab when we do Unit 6 this semester. So there are two different types of bacteria, two different types of cell walls for bacteria. There are those bacteria that we call gram-positive, so they have a positive stain for the Graham stain, and then those that are negative. Gram-positive bacteria have much simpler cell walls that's essentially just a really thick layer of peptidoglycan. That's it. Gram-negative bacteria have a slightly more complicated cell wall. They have less peptidoglycan and they have this extra outer toxic membrane. Right? So let's take a look at the composition of the cell wall of both gram-positive and gram-negative bacteria, and let's walk through the Graham stain. This will give you guys sort of a legs up when you start doing the Graham stain in the lab. Okay, so here we have the two examples of the different kinds of bacteria. So on the left, we have a gram-positive bacterium. On the right, we're going to have the gram-negative bacteria. And if we take a cross-section through each of these cell walls, remember, prokaryotic cells still have a plasma membrane, and just outside of this membrane, they have that cell wall. A gram-positive bacteria, if we take a look at that cross-section, down here's the plasma membrane, so the inside of the cell should be just below it. On the outside of that plasma membrane, you're going to have the cell wall. And for a gram-positive bacteria, this cell wall is a thick layer of peptidoglycan. That's it. Simple. All right? Gram-negative bacteria, on the other hand, here's the plasma membrane down here. Yes, the cell wall is still there, but the composition is a little bit different. In this case, for a gram-negative bacterium, the cell wall has two parts. The first part is going to be a thin layer of peptidoglycan, and that's going to be sandwiched by yet another membrane just on the outside, so on the very, very surface of that bacterial cell. We call that the outer membrane. And if you take a look at the phospholipids on that outer phospholipid bilayer, they have a whole bunch of these little green, they almost look like little green antenna extensions, hairs, whatever you want to call it. These are carbohydrate components that are stuck to the surface of those phospholipids. And it's sometimes these carbohydrates, these extra little components that are added to those phospholipids that make that outer membrane highly toxic to the host organism that this bacterium is invading. All right, so let's talk a little bit about the Graham stain. How does a series of stains differentiate between these two cell walls? Well, a Graham stain technically uses two different stains. It also uses a destain mechanism as well. So let's go through the various steps of the Graham stain. Down below is an example of a slide which has a mixture of both gram-negative and gram-positive bacteria on it. So you can see sort of the difference between those two once the Graham stain has been completed. But if we were to start with this clear slide, it's going to be clear, the cells are going to look clear. The first step of the Graham stain is to add the first stain. And the first stain is called the crystal violet. And this crystal violet is a violet color, maybe bluish in color. And this stain is going to penetrate through the cell wall of both gram-positive and gram-negative bacteria. And it's going to get into the inside of the cell and stain that cell blue. So at this point, all the cells on that slide, no matter whether they're gram-positive, gram-negative, will be stained blue. Okay, the next step is to add iodine. Right? Previously, we've used iodine in lab for detecting starch. Right? We also used it in the eukaryotic microscope cytology lab to stain our plant cells so we can see them under the microscope a little bit clearer. Iodine's really good at staining the nucleus in both the onion and the elodea leaves. This time for the gram stain, we're going to use iodine to sort of capture and retain that blue crystal violet stain on the inside of the cell. So what happens is in the first step, right, we added that crystal violet that went through the cell wall, through the plasma membrane into the inside of the cells. To ensure that that blue stain stays there, we add the iodine, which also goes all the way into the inside of the cell, and now it sort of reacts with that crystal violet stain, and it makes these big aggregates of that stain. So those big chunks of that stain are really difficult. It's really difficult for them to get back out of the cell. So we're helping to retain that blue dye on the inside of the cell by adding the iodine. All right, so far there's no difference between our gram-positive and gram-negative bacteria. And we've added two components. Our first stain, which was a gram, essentially the gram stain, which is that crystal violet, and then we added the iodine to retain that crystal violet on the inside of the cells. The next step is key, and this step is a decolorization step. To decolorize the cells, to attempt to wash out that crystal violet, what we use is ethanol. Right? So we're going to put ethanol onto our slides. And the ethanol, what it does is it will break apart membranes, and it will allow these holes to be made in the membranes so that that crystal violet stain will be able to go through. Here's where we start to differentiate between gram-positive and gram-negative bacteria. Remember, gram-positive bacteria have that really thick peptidoglycan layer. The alcohol really can't do anything to that, right? It can't penetrate through that, and so the gram-positive bacteria are going to stay blue. On the other hand, the gram-negative bacteria, the ethanol is going to degrade that outer membrane. It's going to get through that thin peptidoglycan layer, get into the plasma membrane, poke holes in it, and allow all that blue stain now to leave the gram-negative bacteria. So now it decolorizes those gram-negative bacteria. So if we were to look at the slide down here, now we can imagine gram-positive cells would be blue, the gram-negative cells would be clear. Was it easy to find your clear cheek cells on that slide when you were looking for them? Probably not, right? So you want to add a counter stain to your gram-negative bacteria so that you can actually see them on the slide, because right now they're colorless after the D-stain. So we add our last stain. So far we've only used one, right, the crystal violet. We're going to add our last stain. We call this the counter stain, and this is going to be a stain called safranin. It's kind of a reddish pinkish color. You add the safranin to your slide, and it's going to stain all the cells. Again, it's not picky at all. But the gram-positive cells are still blue, and that blue is pretty deep. And the deep blue is really not going to be much affected by that pink safranin color. So those gram-positive cells are going to remain blue. On the other hand, the gram-negative bacteria, I'm going to have to plug in my computer, the gram-negative bacteria, these guys were clear, right? And now we add that safranin, that red dye. It's going to make all those colorless cells now appear red. Helps us to be able to see it. So down here, you can see the gram-positive cells, those little purple spheres, purple bluish spheres. Those are the gram-positive, and then all the pink rod-shaped cells are going to be the gram-negative cells instead. Okay, before we get to this, I'm going to plug in my computer and we'll finish up this chapter.